Well, good morning, everyone. Thank you for joining the Imagion Biosystems webinar today on November 27th. We're very pleased to be able to have the opportunity to share with you and our story as we're making progress here. If you go to the next slide, please. Joining me on the call today is fellow non-executive director Brett Mitchell, Melanie Leiden, and Ward Detweiler. Apologies for today. They had other things come up that wouldn't allow them to participate. But Brett's on the call, and during the Q&A session, if we have any questions that might be addressed to one of the directors, that's fine. He's happy to participate as well. So if you go to the next slide, I thought I'd just spend a few minutes. I know the majority of you are probably existing shareholders and are familiar with Imagion Biosystems story, but we might have some new shareholders or interested parties. So I'll just take a couple of minutes to sort of run through the products and technologies that we have, the rationale for why we're pursuing this, and then we'll talk a little bit more about where we are currently with our programs, and in particular, what we're looking for coming up at the EGM meeting we have in a couple of weeks. So we consider ourselves a clinical stage biotechnology company. Primarily, we are focused on using our magnetic nanoparticles for imaging, but they also have the potential for drug delivery. I like to walk before I run, so we've been working the company with the idea first of demonstrating that we can develop molecular imaging agents and change the way imaging is used to diagnose cancer. But then in the background, we've been pursuing some intellectual property along the idea of still using our particles for drug delivery. In the imaging space, we have three primary products in development. Our lead product is for the detection of nodal disease in HER2-positive breast cancer. That we'll talk a little bit more about that, but that's the furthest one along in terms of its development. But we have a pipeline of other imaging agents using the same underlying technology that we'll go through that are focused on prostate cancer and ovarian cancer. And you'll see as we go through this sort of why they're different products and how they target different types of cancer individually. So if you go to the next slide, please. I think everybody's familiar probably with what the unmet medical need here in cancer is. We have lots of great ways of screening for cancer. We've got blood-based tests like PSA for prostate cancer or CA-125 for ovarian cancer. We have screening tools like mammograms for breast cancer. But ultimately, none of those screening tools are diagnostic per se. That is, they tell you that you're at risk of some type of cancer, but they can't tell you whether or not specifically you really have the cancer. So ultimately, we end up doing biopsies in order to be able, excuse me, to confirm the presence of the cancer. Imaging is often used as an adjunct in that process, but imaging to date has not been diagnostic. That is, because the imaging modalities that we have today can identify something that is a region of interest. They see a spot, looks abnormal, or maybe it shouldn't be there. But again, here, imaging tools today don't allow us to actually diagnose cancer. And so therein lies the unmet need. How do we help transform imaging from being something that's a helpful tool, but isn't fully diagnostic, and avoiding then being able to avoid unnecessary biopsies? So if you go to the next slide, we have a cartoon that sort of illustrates it, how this is going to work. So in conventional imaging, you'll see an image, as I said, you'll have anatomical information about maybe the size and shape or irregularity of a lesion that you see. But the radiologist, the imaging experts look at that and say, okay, that shouldn't be there, but I don't know exactly what it is. We need to do a biopsy or we need to do some further investigation. Our technology is aimed at changing that. By using our molecular imaging agents, our imaging agents actually bind to the tumor. And then when you see the MRI, what you see is the presence of our particles. And now we know the particles are there only because it's in fact cancer. So we combine that diagnostic utility of a pathology report where we understand the molecular basis of what's there with the imaging basis of being able to see it in vivo in the body. So MagSense imaging agents are designed to be a molecularly targeted imaging agent and therefore enabling what we now refer to as molecular imaging. So if you go to the next slide, we'll see how that's actually works its way out in real human beings. So we use a targeting moiety of some kind, an antibody or peptide, that's highly specific for the type of cancer that we want to try to diagnose. And when our particles are present in an image, an MRI image, they generate a dark or hypo-intense contrast to the image. So what you see here is one patient's lymph nodes as part of our original phase 1 study, where on the left-hand image, you see pre-dose, what those lymph nodes look like before we gave our imaging agent. And then on the right-hand side in that study, we went ahead and did an image after we dosed the patient with our imaging agent. And you can see there's a significant change in the darkening of the nodes. And that darkening is a direct result of the presence of our particle. So in this kind of image, what you see is not only do you have the anatomical aspect of the irregularity of size and shape of those nodes, but now we get this spotty heterogeneous color pattern to them, which is indicative of the presence of our particle. And that amount of color change, that hypointensity change, is quantifiable. So we can use algorithmic solutions such as machine learning or AI to help teach the imaging machines how to look for this spotty pattern and identify the fact that our imaging agents are there, and our imaging agents are there because we've used a targeting moiety of some kind. So if you go to the next slide, we can chat a little bit about why this is going to be transformative, right? So first and foremost is the fact that we're using existing MRI-based technology. We're not introducing a new way to try to image. We're taking advantage of the fact that there are a large installed base of MRI machines around the world. We're all probably familiar with having had an MRI for one reason or another, but typically MRI has not been used as a primary tool in cancer. And that's because, again, you see a lesion, you see a spot, but you don't really know what it is. So we think that we can help improve the way MRI is used specifically in oncology, because we have the advantage then that our particles are highly specific. We're not just trying to generically look for things, do a whole body scan and just see if something abnormal comes up, but we're using highly specific targeting moieties to make sure that the cancer we're looking for is identifiable. It's clearly a safer alternative than using, for example, PET tracers, which use radioactivity, or even mammograms or X-rays, which use ionizing radiation. So combining the fact that we can use existing technology with specific outcomes and in a safer mode, we think that we ultimately can get to earlier detection as well. And the fact is that we can apply this across a wide range of cancers. So it's in fact a platform technology, and you'll see as we look at our pipeline how that's starting to play out. So if you go one more slide forward, you can see a little bit that we've now already completed a phase 1 study. Our phase 1 study was for, as I said, HER2 breast cancer. But here we were looking for nodal disease. So that example that you saw earlier of a patient's lymph nodes. We're trying to detect whether or not the patient's primary tumor has begun to spread, and typically it spreads first through the lymph nodes. So our test is designed to aid the surgeons and the oncologists in identifying, as soon as a patient's been identified as having the cancer, can we in fact determine how extensive that is? Is it an early stage without it having spread, or can we non-invasively determine whether or not there are nodes involved? So we completed a phase 1 study. We did a blinded independent radiological review so that we knew that what we were looking at actually had merit. And now our plan is to try to move forward with that. If you're not familiar in the drug development and imaging agent kind of development, you first do a safety study like we did in phase 1, and then you begin to progress through a phase 2 and a phase 3, which is when you begin to prove your case that in fact you can, the ultimate claims that you can make in terms of its diagnostic utility. So if we go on to the next slide, we can now start to see how that starts to place out in terms of the platform and its applicability to multiple types of cancer. So our lead product, as I said, is for the identification of nodal disease in HER2 positive breast cancer. It's also important to understand that in breast cancer, HER2 positive is one of the more aggressive forms of breast cancer. And so the reason that we've decided to tackle that first is the fact that it's an aggressive form of cancer. It often metastasizes early in patients, and there's a known course of treatment for it. So if we can help the surgeons understand whether or not there's nodal metastasis, then the course of treatment might be modified in favor for the patient. So we still see that as being a very valuable unmet medical need, even though it's restricted only to one type of breast cancer. But we've now established particles that can also detect prostate cancer and ovarian cancer. And here we're going to move further forward in the diagnostic regime. So unlike the breast cancer application where we're waiting to look at nodal metastasis, for both prostate and ovarian cancer, we believe we have the opportunity to do primary tumor diagnosis. So today, a man gets a test for PSA in his blood. If it's positive, are elevated, they want to then typically do either a PSMA PET test using a radio tracer, or they'll go straight to a biopsy. We have about 1 million men in the United States annually that have biopsies, of which about 20% of them are negative, and another percentage are false positives. So here, if we could avoid doing biopsies on those patients and being able to identify right after an elevated PSA test by a simple non-invasive imaging test whether or not you have prostate cancer, because you'll see that hypo-intense image contrast from the presence of our particles, we think we have a way to really affect how prostate cancer might get diagnosed in the future. And then, of course, the poster child from my perspective, if you will, of being able to do early detection is ovarian cancer. For those that are not familiar, ovarian cancer is known as the silent killer because today we don't have a very good way of detecting ovarian cancer. The blood test for CA-125 is not specific for ovarian cancer, and so they do what's known as watchful waiting by ultrasound. And typically, ultrasound doesn't see an ovarian tumor until it's a stage 3 or stage 4. And the data are very clear that if you can detect ovarian cancer as early as stage 1 or 2, the success or outcomes rate is much better, typically above 60 to 80%. But if you don't detect ovarian cancer until later stages 3 or 4, then the survival rate goes down close to 10 or 20%. So this is a good example where if we can develop an ovarian cancer non-invasive test and avoid that sort of non-specific and non-sensitive method of ultrasound, we think we have a way to really affect the detection of ovarian cancer and the outcomes for patients. So all these products use the same underlying technology. And so that's why we consider the fact that we have a platform to be able to apply our imaging technology to a variety of different types of cancers, so long as we have a targeting moiety, an antibody or peptide of some kind, to target those types of cancers. So if you go to the next slide, we're going to spend a few minutes now talking a little bit more about the rationale for why we're pursuing this. We're the first company to ever try to develop a molecular MRI imaging agent. Imaging agents are already in use. So some of you may be familiar with something called gadolinium and had a contrast-enhanced MRI, for example. But they are non-specific. They help enhance the image, but they are not specific and don't tell you if what you have is cancer, for example, in oncology. And those imaging agents are used for quite a variety of things. Gadolinium is also used for vascular disease by looking at the flow of blood because it improves the image contrast associated with the blood versus the vascular walls. So the existing imaging agents are not specific for oncology applications. But it's important because there are 50,000 MRI scanners around the world. There's probably 5 times as many MRI scanners as there are PET imaging systems. And so we see the opportunity to serve and expand access for patients around the world, because there are whole parts of the world that really don't have access to technology like PET, but have access to something as simple as an MRI. And so the fact that imaging agents in general are already known to be used, and that there's a large installed base of MRI imaging agents, really we saw as the market opportunity for us. And so the trick then was, how do we develop that molecular aspect of things? And what you saw, I think, previously in the slides is that we've come a long way from the concept to actually now having clinical data and a pipeline of products that we can bring forth. We've started to look at the health economics of how would our product fit, right? So we know it fits within the clinical workflow, but how would it really fit from an overall commercial perspective? Because clearly we want to be able to develop a product that will have commercial traction and be able to be profitable. And so that's often affected by whether or not the economics in the healthcare system make any sense. And so we started to do some of that work across each of those different types of products. And what you see on the graph on the right is a little bit of the homework showing the example, particularly in breast cancer. So that for every patient that would be normally treated by the standard of care, if instead the standard of care became to use our MagSense technology, we would save money compared to the existing standard of care for every single patient. And that includes the cost of having administered the imaging agent. So we build into the right-hand column there the cost of doing our test versus the cost compared to existing. So we know going into all of these that we have an economic advantage to the healthcare system. We've got a better advantage in terms of the answer that the radiologist gets. and we have a large installed base to be able to bring our technology to. So we think that this makes a great rationale for why we want to be first in line to develop these molecular imaging agents. If you go to the next slide, then we'll go a little bit further about, well, where does that lead us to from an overall company? I've said for a long time now that our overall goal is not to try to be a global powerhouse in terms of from a commercial perspective, but to identify strategic partners that could help us commercialize this. We think it's more likely that we would get traction if we had a large strategic partner who's already in that imaging space. And so there's probably 40 to 50 companies that are well known that have either imaging equipment or already sell contrast media, et cetera. So we know that there are global players that have the infrastructure that would help bring our product to market, and that are already have products that are either complementary, but since nobody has what we have, we're the first to be able to bring it to them. And so there's an element there where we need to be able to effectively de-risk our technology and demonstrate to them that it will have commercial legs, but we're not bringing them something that they haven't seen before. So all of these companies have seen various types of contrast media and that. And so we think that right now we're in that period of time in the growth of our company and value of our company where we're bridging the early R&D stages to where the value is going to start to be generated because the maturity that we're developing in our products. And if we take a look at the next slide, you'll see how that starts to translate out in terms of strategic relationships. So here's just a couple of examples from the marketplace relatively recently where companies, either the acquirer, and that acquisition could be an outright M&A activity, or it could be an investment to further help develop. For example, Lantheus's investment in radiopharma was not an outright acquisition, but a strategic investment to help them develop. But most recently this year, Hologic acquired Endomagnetics, another iron oxide nanoparticle company, not doing molecularly targeted imaging like we are doing, but a company that's developing iron oxide particles for diagnostic purposes. And we see the valuation that they got there. So we think that as we continue to develop our portfolio, that we will have an opportunity to be on the radar screen to try to attract these partners, perhaps not necessarily as an outright M&A activity, but to help us fund and further develop and then commercialize the technology so that we have the success there. And so if you go to the next slide, we'll see where we are then with regard to sort of the path for our growth and value proposition. So as I said earlier, we have the first product already completed phase 1. That was last November. We finished the phase 1 study. We closed that study out. And we have 2 other products that have made it through what we would refer to as the preclinical stage, where all the evidence associated with the efficacy, prospective efficacy, specificity, sensitivity, et cetera, have been worked out. And now what we need to do is we need to move that first one into phase 2. And the first step in doing that is to achieve what's known as an IND, or an investigational new drug application with the FDA, because we did the phase 1 study in Australia, but we know in order for us to actually get in front of the FDA and get access to a larger patient population, we need to be able to do that in the United States and in Europe. And the best way to do that is to get an IND clearance from the FDA. So our goal right now, our nearest term goal, is to try to file that IND and then begin that phase 2 study. And in that phase 2 study, we'll be looking to optimize the dose in the imaging protocol so that eventually when we get in the phase 3 study, we've got everything locked down. It's exactly as it would be in the true clinical setting that we go further test that in a phase 3. But in the meantime, we also want to try to progress the other 2 products, because we know that if we can get those IND enabling studies and in fact file an IND with both of those, we now make ourselves more attractive to those strategic partners, because we're not a one-trick pony, so to speak, with HER2 breast cancer, but we clearly have demonstrated we got a portfolio of products that are all ready and able to be in the clinic. And we think that if you looked at what was important aspects of those strategic partnerships, is the maturity and the de-risking, if you will, of the technology. Strategic partners see the progression through the clinical development as the de-risking of the technology. And so our main goal right now is to keep moving this R&D pipeline along, because every step that we take along there adds value and adds attractiveness, if you will, to those potential strategic partners. So if we go to the last slide, just an update on our current status. So hopefully everyone is aware that we have a shareholder meeting coming up on December 9th. That meeting is to be held at 10:30 a.m. AEDT. There was a notice of meeting provided on November 7th. If you have not received that, you can go to our website under the Investor Hub and the ASX announcements, and you'll see from November 7th that notice of meeting. So please, if you're an existing shareholder, please plan to attend that meeting. There's a few key objectives of that general meeting. The first and foremost of which is to approve the issuance of the shares to complete a $3 million capital raise. The information about that capital raise was also available on our website. But the key thing here is that we are seeking shareholder approval to complete that capital raise. There's a few other resolutions that will be there, but they are all related to capital raise and/or reducing the debt through the issuance of shares. And so we are seeking shareholder approval effectively to complete all of that, and so that we can finish the year with cash in the bank and a reduction of our debts. So we'll have time at the end of this for some Q&A if there are any questions about the specifics of that meeting, but want to be sure that everybody's aware that that's coming up. On a more general basis, in terms of current status and priorities, the key thing that we've identified, as I said, is that we really now, with the funds, assuming that those funds get approved at that meeting, then the next thing for us to do is really begin to reapply ourselves to our R&D pipeline, the first and foremost of which is filing the IND for the phase 2 study. We've got probably about 6 to 9 months worth of work left to be done in order to get to that phase, to get to that IND. So our goal is to be first half of next year, get that finished up, get that IND filed, and be preparing to initiate that phase 2 study. We've already started to organize ourselves around that. You may have seen in our most recent business update that we've now brought on a medical affairs advisor, very experienced woman, a radiologist previously at Johns Hopkins University in breast imaging, but also spent a number of years at Hologic, one of the largest manufacturers of mammogram or mammography equipment. She was their head of medical affairs. She recently retired, but has taken an appointment with us to be our medical affairs advisor. She'll be helping us complete the design of the study and make sure that the study is going to get us ultimately what we want. And we couldn't ask for anybody more experienced than somebody who comes with her credentials. And we've already started to line up a couple of the other key things involved in getting that IND filed. That includes we need to manufacture a batch of material to be used in the study, get our documentation in order. So we started to pull the team together, and assuming that we get our capital raise approved and the funding in, we'll begin to really push towards that objective of getting that IND filed. And then, as I said previously, we will also start to work on our prostate and ovarian cancer, trying to move them towards that IND enabling work eventually. But we're going to be a little bit careful, making sure $3 million will get us so far with the IND and a little bit with the prostate and ovarian cancer. But I want to make sure, one of the things we committed to earlier in the year is we're going to only spend as we go and not get over our skis too much. And so we will try to advance those programs in line with what the availability of our funding is. Just to make folks aware, we've got a couple of upcoming events that we'll be participating in on Monday through Wednesday of next week. Ward Detweiler and I will be at the Radiological Society of North America in Chicago. This is probably one of the world's largest meetings for imaging companies. It's a chance for us to get some meetings with some of the key players that are part of that potential strategic partnering. Those that have been long-term shareholders are aware of the fact that we've had a collaborative relationship with Siemens Healthineers. They help support our phase 1 study and have an expanded arrangement with them for them to help support our phase 2 study. Since that had been put on hold for a little while, we've been waiting to get that restarted. And in fact, next week we'll be meeting with them at the RSNA to sort of kickstart that relationship as we move towards that IND filing and the phase 2 study. But of course, we'll be looking to develop other relationships. There are a number of other companies that are that will be critical for us to be able to have the support during that phase 1, our phase 2 study, for example, and also beginning to look at some of the other contrast media companies that might be interested in what we have. And then there's also the San Antonio Breast Cancer Symposium coming up. Again, existing shareholders may be familiar with the fact that we presented at that meeting a number of times. And in fact, it was last year at this time that we presented the results of our phase 1 study. So we'll be back there. That's a big opportunity for us to get in front of some of the potential clinical site investigators that might be able to participate in phase 2, because that meeting's really more about clinical applications. So a number of breast surgeons, for example, MedOncs, will be at that meeting. So between the imaging meeting at Chicago and the San Antonio Breast Symposium, we've got coming up a couple of good opportunities to get ourselves in front of the kinds of folks that will be instrumental in helping us to continue to advance our programs. And so with that, I will go ahead and open it up for questions. I'll remind everyone, because I didn't at the beginning, we did not lodge this presentation with the ASX, as it's largely identical to previously lodged presentations, but we will make it available on our website. So you can go to our website under the Investor Hub, and you'll be able to find it there. Or if for any reason you're unable to find it, you can go ahead and contact us at info@imagionbio.com, and we'll be happy to send it along. And with that, I will go ahead and open the floor for questions.
Hi, Bob. Just confirming that we've received no questions as yet.
Okay. All right. That's great. Well, I appreciate everyone attending the meeting today. If you have any questions afterwards, please feel free to go ahead and send those to that info@imagionbiosystems.com email address. We'll make sure we get back to you on that. If you're an existing shareholder and have not yet voted for your shares for the EGM coming up, please be sure to do so. You're also eligible to participate in the EGM directly and vote your shares on the day. But please, make sure that if you're an existing shareholder, you do that. And if you're a prospective new shareholder and interested in making an investment in the company, we'd love to hear from you, and hope you continue to follow us. So thank you very much for attending our webinar today.